division of one cell to two and differs between prokaryotic and eukaryotic cells. Shown here is binary fission, the process by which prokaryotic cells asexually divide. Eukaryotic cells first undergo interphase and then mitosis. Approximately 70% of the time a eukaryotic cell is undergoing a mitotic division, it is spent in interphase. Interphase is broken up into three subdivisions, G1, S, and G2. During time in G1, organelles in the cells are duplicated. Essential enzymes are also synthesized during G1. The S phase is when DNA synthesizes and replication occurs. In G2, the cell continues to grow and divide. After interphase, the eukaryotic cell enters the M phase. There are many checkpoints in the cell cycle, represented here by the red arrows, that use both cyclin and cyclin-dependent kinases, which will halt the growth and development of a cell in preparation for division. If a checkpoint decides that the cell is not strong enough to divide, it will begin to circulate through G0, which is simply a period of rest. At the end of mitosis, usually with telophase, cytokinesis occurs. The process of cytokinesis splits the cytoplasm and essentially creates the two new daughter cells with identical numbers of organelles and the same concentration of cytosol. Animal cells divide at a location known as the cleavage furrow. Cytokinesis in a plant cell occurs in a region called the cell plate. Mitosis, or the division of cells, is essential to life. Cells are constantly renewing themselves to ensure that structure is optimal to function and that function is at its peak capabilities. Cells have mechanisms to destroy themselves with lysosomes and peroxisomes, and the process of mitosis allows more cells to then be created. Organisms do not grow because individual cells grow larger. Multi-billion-celled organisms, like humans, are able to grow because the number of cells can increase through mitosis. There are four main phases of mitosis and anaphase, followed by the previously discussed cytokinesis. In prophase, chromatin starts to condense into distinguishable chromosomes, as represented here by Twizzlers. Nucleoli disappears and the nuclear membrane starts to break down. Centrioles, represented by Kit Kats, move towards opposite ends of the pole, and chromosomes move into the center. The most notable element of metaphase is the alignment of pairs of homologous chromosomes along the metaphase plane, which is an imaginary line in the middle of the cell between the poles established by the centrioles. The mitotic spindle begins to form, and the arms of the spindle are based on the centriole towards the appropriate pole. Centrioles also release astral rays, which guide the spindle fibers. The chromosomes attach themselves to the outside spindles. They would appear to be in a circular arrangement viewed from the point of one of the centrioles. The double-stranded chromosomes are attached to the spindle fibers by centromeres. Anaphase begins when sister chromatids separate when the centromere separates. Each sister chromatid is now known as an individual chromosome. Therefore, the chromosome number doubles from metaphase to anaphase. Spindle microtubules attached to the centromeres that hold the newly separated chromosomes on the spindles pull the new chromosomes towards the poles with the centrioles. Although rare, mistakes in mitosis can be made when a separated chromosome is not attached to a centromere. In telophase, chromosomes are pulled towards the centrioles, and the regions near the centrioles ultimately become the nucleus. As previously discussed, cytokinesis occurs after telophase. Therefore, mitosis creates two diploid daughter cells with nuclei identical to those of their parent cell. Meiosis is the process of halving genetic material so the amount of nuclear material stays consistent from one generation to the next. Gametes from two parents fuse to form a fertilized egg known as a zygote. Meiosis occurs in two phases. Meiosis 1 separates homologous chromosomes, and Meiosis 2 separates sister chromatids. Meiosis 1 is preceded by interphase, where the cell goes through G1, S, and G2 phases. In prophase 1, homologous chromosomes pair and exchange segments. A set of homologous chromosomes is referred to as a tetrad. Chromosomes have points of crossing over, called chiasmata. At these points, genetic material is exchanged, creating a unique combination of traits for the new chromosome. In metaphase 1, the tetrads line up along the metaphase plate of the cell, similar to the process in mitosis. The centromeres hold the tetrads together with kinetochores attached to the centromeres. The microtubules coming from the centriole are attached to the tetrads by the kinetochore. In anaphase 1, homologous chromosomes separate, but sister chromosomes remain attached. Therefore, in telophase 1 and cytokinesis, two haploid cells are formed. Chromosome numbers are still double. Telophase occurs in the same way it does in mitosis, and cytokinesis occurs with the cleavage furrow, as in all eukaryotics. Meiosis 2 is very similar to mitosis, and that is the separation of sister chromatids. Meiosis 2 does not include interphase, though, because sister chromatids in the cells are the result of meiosis 1. Prophase 2 is simply the reformation of a mitotic spindle, coming from the centrioles moving towards opposite poles of the cells. The chromosomes are already condensed, with two sister chromatids attached together. Metaphase 2 shows the lining up of sister chromatids along the metaphase plate, attached to microtubules by kinetochores. Anaphase 2 shows the separation in the sister chromatids, each being pulled along a microtubule towards one of the poles. Telophase 2 and cytokinesis create four haploid cells, each containing a single chromosome. Meiosis, therefore, creates four haploid cells, each containing single chromosomes. processes of meiosis and mitosis are very different, yet essential for the growth and development of any organism.